Well, good morning, everybody. As Craig said, my name's Darren. I firstly like to pay my respects to the Gadigal people as the traditional owners of the country we're meeting on today. Where I live and work in North Queensland is on the traditional country and lands of the Yirraganji and the Jabbergai people, and I pay my respects to them and to all First Nations people in the audience today. So I'm going to talk a little bit about a small pocket of northeastern Australia, which we call the Wet Tropics World Heritage Area. It sits within a broader bioregion, the Wet Tropics bioregion. Now, this is an incredibly important slice of Australia's biodiversity and landscape. It's an incredibly beautiful landscape. It's almost all tropical rainforest. It's irreplaceable in every sense of the word. There was a numerical study that was done on all 173,000 protected areas globally, published about 10 years ago in Science. The Wet Tropics World Heritage Area ranked 6th of all protected areas globally in species irreplaceability. So by that I mean those species in the wet tropics are not captured in any other protected area anywhere. On the basis so in terms of World Heritage properties, the Wet Tropics World Heritage Area ranked 2nd globally. So this is an incredibly important area. Of other World Heritage properties in Australia, Kakadu, which will be known to many of you, came in at number 63. And the Great Barrier Reef came in at number 1,229. So we're talking a superlatively important area. It's also the home of many ancient and endemic plant lineages. 12 of the 19 globally ancient flowering plant lineages are represented in the wet tropics. There's over 700 endemic species. So 17% of the wet tropics flora is endemic at species level. 350 plant species are threatened, either nationally or at state level. That's about 8% of the flora and about 33% of Queensland's total. In terms of Myrtaceae, we've got about 123 species in 31 genera. That's 70% of Australia's rainforest Myrtaceae, and of course the rainforest Myrtaceae are the ones that are most heavily impacted by myrtle rust because of the conditions in which they occur, warm and wet. 60 of those 123 species are endemic to the World Heritage Area, and there are 4 endemic genera. 17 of those Myrtaceae are threatened. The Wet Tropics Management Authority, which is the legislative authority to look after the Wet Tropics World Heritage Area, recognises myrtle rust as one of the 3 principal disease threats to the World Heritage values of this important area. The other 2 are the chytrid fungus, which impacts the frogs and the other amphibians, and another disease of flora, which is Phytophthora root rot. What do we currently know about myrtle rust impacts in the wet tropics? And the answer, sadly, is not an awful lot. There's only been one systematic survey that's been undertaken, to my knowledge, of myrtle rust impacts in the wet tropics. That was done by Rod Fensham, Julian Radford-Smith and Tegan Collingwood in 2021. They surveyed hundreds of sites across Queensland. They categorised species into Category X and Category Y based on whether they're likely to become extinct within one generation because they're effectively not reproducing. So there's Category X species, are those species they figured would be gone within a generation. In North Queensland, we've got a number of those. Backhausia hughesii, it's a wet tropics endemic. It continues its severe decline. We've got Gossia hillii, it's a widespread species, but you can see from the map on the right there that the northern populations are geographically disjunct from the main the main strength of the species, which is down in the southeastern corner of Queensland. Gossia lewicensis was a wet tropics endemic for the uplands of the wet tropics. Again, continued severe decline has been observed recently. Lenwebia lasiaclata, again, that's a widespread species, but the map down towards the bottom right there shows that the wet tropics populations are a long way geographically from the rest of the species. Rhodamnia arenaria, found only on Cape York Peninsula. Rhodamnia spongiosa, widespread again, but again heavily impacted across its range, and Ristantia pachyperma, which is a very large tree with very large leaves of lowland swamp forests. Only one of those species is currently listed as threatened at either state or federal level. What are we doing about it? Well, sad to say, we haven't had the capacity to really stand up much serious activity in northeast Queensland. I'll get to the reasons for that a little bit later in the talk. But what we are starting to do is to work with QDAF, with Jeff and others to launch projects that are focused on species that have the capacity to convert ecosystems. So we've got a lot of individual species that are part of rainforest ecosystems but are not necessarily dominant. But Tristaniopsis exiliflora is one of those species that has the capacity to cause some serious damage and convert ecosystems if it were lost from the landscape, because this species lines the waterways of most of the east-flowing streams in the wet tropics, particularly the steep, rocky, fast-flowing waterways, and it holds the stream banks together. If this species were to be lost from this landscape, it's highly likely you get mobilisation of sediments and all sorts of things along those fast-flowing streams. They all drain into the Great Barrier Reef Lagoon, and siltation and sedimentation are a serious problem recognised for a long time, a serious threat to the World Heritage values of the Great Barrier Reef. So Fensham and Radford Smith gave this one a damage score based on their observations in 2021 of only 2.2, which is relatively low in the scale. But we have seen a lot of serious dieback in this species, but it's very patchy. And we really need to know why that is, whether there are biological correlates, where environmental correlates are these abiotic or biotic factors that are driving this patchiness. So that'll be the focus of this project. Other current activities, so there's a fair bit of species rescue and ex situ conservation going on, but this is all entirely unfunded. And let me talk to you about a wonderful bloke who's a colleague of mine at James Cook University. His name's Brandon Espy. Some of you will have had contact with him already. Brandon's an environmental officer with the estate team, so they manage the campus estate. And Brandon, since about 2019, has been mostly in his spare time on weekends, going out and trying to secure as many North Queensland threatened species, not just Myrtaceae, but any threatened species, and bring them onto the campus, the Townsville campus of James Cook University. We've got two main campuses, one in Cairns where I am, which is in the midst of the wet tropics, one in Townsville where Brandon is, which is just at the bottom of the wet tropics in what we call the dry tropics, where the rainfall is about half of what it is in Cairns. And so Brandon's been systematically building their ex situ conservation collections there. He's got, at time of writing, over 36 species known to be impacted by myrtle rust in that ex situ conservation collection. He's got multiple genotypes of many of them, if not most of them. And he's been able to observe for some of those that there's quite a lot of variation in the susceptibility in situ on campus to myrtle rust. So I think he's built by design, but partly by accident, but mostly by design, a really important ex situ collection that we can use for observations over a longer term, but also for research. Some of the key species he's got in that collection are listed there. The Backhausia tetraptera is a really interesting one. This is a species that was only discovered about a decade ago, just on the hill, literally behind the Townsville campus of JCU, and people hadn't realised that this tree that was growing down this gully, you could almost see it from the botany department of JCU Townsville. But it looks very, very much like a common and widespread Gossia species, except when it's flowering, and so it hadn't been noticed for a very long period of time. It's still only known from that one population along that one stream, plus a couple of other isolated trees elsewhere. It's critically endangered. It was listed as critically endangered based on its restricted distribution before myrtle rust, We thought it was going to escape, but Brandon tells me just in the last couple of weeks he's seen myrtle rust on this species in the wild. So it's a concern. The other thing we would like to do, we haven't yet initiated this project yet, but we've built a collaborative partnership involving many people in this room and organisations that they represent. And what we wanted to do was to establish a program to drive ex situ conservation across multiple sites down the east coast of Australia for the North's most critically endangered myrtle rust species. We modelled this project, which we called Rust Proofing the Rainforest, on another ex situ conservation project we've been running for 5 years now, which has been focused on the climate-threatened species of the mountaintops of the wet tropics. So we've picked up that model, and we're hoping to apply that to myrtle rust. And this involves multiple components. There's seed banking research, there's propagation in botanic gardens of varying genotypes, there's genetic analysis to ensure that the ex situ collections reflect the wild diversity to the extent that that's possible, and then the disease susceptibility research. Ultimately, the point is to build a functional, representative ex situ conservation collection to secure these endangered species. Running out of time, so I'll move quickly through this. In brief, as I said, it'll be collecting and propagating species, securing them in multiple botanic gardens along a climate gradient down the east coast of Australia, assessing the genetic diversity, understanding disease incidence and resistance, and all of the rest of it. Whole range of partners, both here in Australia and the east coast, and also through Heidi, Dungey, Scion in New Zealand. This is an indicative partnership list. As I say, the project hasn't been funded. We've been unsuccessful with a couple of attempts to date, so this is indicative only. What are the challenges? In the wet tropics, we've got 2 million hectares, the wet tropics in Cape York, over 2 million hectares of rainforest. It's all highly suitable for myrtle rust infection. The mean annual temperatures in Cairns range between a minimum of 21 and a maximum of 29. It's right in the sweet spot for myrtle rust. And most of that 2 million hectares is highly inaccessible. There's hardly any roads of any type or any access of any type over much of that landscape. Large and diverse Myrtaceae flora. As you all know, and everyone struggles with this, canopies can be difficult to observe when they're tall and they're closed. We have very limited local capacity to really do much in this space. We have 2 small university campuses in Cairns, which is the major population centre in the region. Cairns is about 120,000 people. It's a large town. We've got a very low population density across that whole extent of northeast Queensland's rainforests, and we're a long way from capital cities. And you know, there's a negative correlation between, it's an isolation by distance model when it comes to political influence. If you're a long way from the major population centres where political decisions are made, you're relatively invisible and you've got a much reduced capacity to influence. So that is a continuing challenge. What are our needs? We need to co-design whatever we do with rainforest Aboriginal peoples. They've managed those landscapes for tens of thousands of years. They understand how to work in those environments. We must work with them to ensure not only what we do is effective, but is culturally safe. And that includes training Indigenous rangers in myrtle rust detection and monitoring, all those sorts of things. And Fiona and Jeff and I have already had discussions about bringing those programs that have been operating in the south to the north. We need to be deeply engaging with not only the land management authorities, the Queensland Parks and Wildlife Service, the Wet Tropics Management Authority, but also rainforest restoration practitioners. There's a lot of activity, NGO activity in the north that's seeking to reconnect fragmented parts of the landscape. They're planting a whole bunch of Myrtaceae species. The long-term viability of those restoration activities is going to be maximised if we can supply them with resistant material. Germplasm capture, there's an increasing role for tropical botanic gardens, and I'm going to make a pitch here for greater recognition of university campuses, not only in regional areas, but across Australia and New Zealand as functional ex situ conservation sites. Many of them behave exactly like botanic gardens. They do all the sorts of things botanic gardens do. They're managed like botanic gardens. I suggest we should be recognising them as botanic gardens and bringing them into discussions and representative bodies that deal with the role of botanic gardens. University herbaria already have a seat on the peak bodies, such as the Council of Heads of Australasian Herbaria. I think we should be thinking about the same thing for the university botanic gardens as having a seat on Charbag. We need to collaborate to increase our capacity. As I say, we've got restricted capacity in the north, but we're very good at talking to people. We're mostly pretty friendly. So if you have a project that needs a tropical site to test an activity, then come and talk to us. Whole lot of collaborators on this, and thank you to all of those. And thank you.
I might just say something. I'm not really sure if it's a question. I'm just thinking about our near neighbours, the Torres Strait Islands and the PNG and these areas. I mean, you know, we've got these vast areas that you're sort of talking about in North Queensland, and then there's just beyond. And I feel like we're really not even looking at the moment at what's going on there. Do you have any comments? That's all.
Yeah, so we interface with Southeast Asia. We are the biosecurity front, if you like, for the natural distribution of pathogens and weeds and all of those sorts of things. All the other coasts of Australia, really the pathways for biosecurity incursions are really through people, through travel, through ships, through planes. But in the north, we interface naturally with Southeast Asia. So we have the natural migration of people across the Torres Strait. You know, the closest Australian island to New Guinea is within sight of the coast of New Guinea. So we get natural movement of people back and forth onto Australian territory to access Australian health services. Airborne pathogens like myrtle rust and potentially others are just going to blow straight over from New Guinea onto Australian shores and step across the islands of the Torres Strait and come into northeast Queensland. So, you know, the Australian government recognises this. There's been a lot of investment into biosecurity surveillance and monitoring in Northern Australia. Some of the federal DAF team with the Northern Australian Quarantine Strategy routinely visit sites in the southwestern Pacific and in Papua New Guinea and specifically look for the early incursions of diseases that have come down through the Indonesian archipelago or whatever. So there's a lot of work going on, but we're not doing enough in terms of monitoring and understanding the pathogens that are already here.
Yeah.
Thanks. Another one over the back.
Becky. One thing I've been thinking about, and with the earlier stuff with New South Wales and Queensland as well, is has any kind of remote sensing work being done, and is it being done? And that was kind of linking back to thinking about carbon yesterday, because with some of those damages, maybe not up in the north of Queensland, but we could probably quantify using remote sensing where the damage is really severe. Yeah.
Yeah, and that's why I was really interested in a couple of those talks yesterday that we're looking at, you know, capturing spores from airstreams and also remote sensing for, you know, thermal imaging and hyperspectral imaging. And I think I asked a question about, you know, to what extent can we upscale thermal imaging and hyperspectral imaging technology from a nursery environment to airborne systems. I'd love that to be possible. It may, of course, be just technologically too challenging, and it may be that those signals are not coming through strongly enough off those rainforest canopies for the detections to be reliable. But it's the only feasible approach to intensively monitor for for canopy outbreaks across a landscape such as the one we're looking at in northeast Queensland and Cape York.
We've in Pine, we've had quite good success with high resolution satellite imagery, looking at larger scale kind of outbreaks, and that's available. It's banked, so you can kind of go and look and see what's there or whatever. And that's kind of just based on RGB, I think. So we don't necessarily need, for high level things, I don't think we need the hyperspec.
I might just jump in, Craig, that's right. I think it's going to be, Pine is a much easier system to work in. One of the biggest issues that we have and we've looked at eucalypts, is what you're seeing here is you've got all this red flush, which sort of occurs a certain time of the year, but not always exactly the same time and not across the whole landscape. So you're picking up the type of symptoms that we'd be picking up or looking for myrtle rust. It's very difficult to tease that out as myrtle rust or just flush.
And there's no myrtle rust there.
Yes.
So when you get to high altitudes, many plants produce this red flush, which is anthocyanins, which are sunscreens. So it's not a response to myrtle rust. So there's a real challenge in differentiating, natural growth responses to the other environmental challenges in this landscape from a myrtle rust infection. Yeah.
I was just wondering if you could use canopy death as a proxy rather than a myrtle rust itself. I think that's what Stuart was trying to say. So you could maybe use broad spectrum LiDAR to do that instead.
Yeah.
Until we figure out how to do it.
Absolutely. I mean, there are other. So Phytophthora was another thing that I mentioned is on the radar. Sorry about the pun. For monitoring, and it produces patches of dieback which probably look pretty similar from a satellite to patches of dieback of myrtle rust. But so there's probably some work to do there. But I agree that's promising.